Hello, my name is Sebastián Castaño Pesante, and I will be presenting to you the angiotensin II stimulation in microglial cell pro-inflammatory response, the role in multiple sclerosis. This study took place in the Department of Biochemistry and Pharmacology in San Juan Bautista School of Medicine in Caguas, Puerto Rico, and it was supervised by Yaritza Inostroza Nieves. What are our hypotheses and aims? Well, this study aims to demonstrate that high levels of angiotensin II contribute to the progression of multiple sclerosis by measuring the pro-inflammatory activation of microglial cells in vitro. In other words, we expected that high levels of angiotensin II would activate the pro-inflammatory response in microglial cells. Why are we doing this? Well, to this day, the pathophysiology of multiple sclerosis remains a mystery. This complicates the treatment and prevention of the disease, so in order to develop better pharmacotherapy and preventive measures for future patients, the pathophysiology must be clear and well studied. And although this study will not unravel the whole mystery, it is a step in the right direction. As background, angiotensin II is the principal effector of the renin-angiotensin-aldosterone system, better known as RAS. This has well-documented effects on the cardiovascular and renal system, mainly by the interaction of angiotensin II with the angiotensin II type 1 receptor, or AT1R. The regulated effects of angiotensin II lead to hypertension and cardiovascular disease. Increasing evidence suggests that angiotensin II may also play a role in the pathophysiology of neurodegenerative diseases, such as multiple sclerosis. There have been many findings of increased AT1R expression in the inflamed brain on astrocytes, microglial cells, and neurons in multiple sclerosis patients. Studies with experimental autoimmune encephalitis mice have shown that angiotensin II concentration were greatly increased in these mice, while treatment with angiotensin II inhibitors ameliorate the EAE onset. In addition, there seems to be a crucial role in the acute setting of the neural injury with the production of interleukin-6, tissue growth factor beta, T helper 17 cells, and the activation of the NF-kappa B pathway. Our methodology was divided in multiple steps. In order to study the angiotensin II, AT1R, and AT2R levels in multiple sclerosis, we used C57BL/6 mice with or without EAE and measured the brain angiotensin II and AT1R levels by quantitative polymerase chain reaction, or qPCR. To investigate angiotensin's role in microglial activation, human microglial cells 3, or HMC3, were treated with angiotensin II in presence or absence of an AT1R antagonist, losartan. The expression of the inducible form of nitric oxide synthase, or INOS, was measured using real-time PCR. IL-6 and tumor necrosis factor alpha levels were measured using ELISA. Nitric oxide production was measured using the Grice reagent, and reactive oxygen species production was measured using the Muse oxidative stress kit. The results were as following. In EAE mice, angiotensin II and AT1R gene expression in brain tissue were significantly increased when compared to control mice. Angiotensin II had a 3.2-fold increase, while AT1R gene expression had a 2.6-fold increase. ATR2 expression was not significantly increased, and INOS relative gene expression was significantly increased, as you will see in the next slide. Angiotensin II treatment upregulated INOS mRNA expression in EAE mice compared to the control counterparts. There was a 3.4-fold increase, while also inducing INOS overexpression. Nitric oxide production was significantly increased, and treatment with losartan blunted its production. In HMC3 cells, treatment with angiotensin II upregulated the gene expression of IL-6 by 3.9 folds and increased the IL-6 concentration by 83%. The changes in IL-6 were blocked by the AT1R antagonist losartan, as you can see in the graphs on the left. In addition, ROS production was significantly increased by angiotensin II, and treatment with losartan blunted its production, as we can see on the left. In the top, we have a flow cytometry, where green and blue, which is the M1, is an anti-inflammatory response, and red is M2, which is a pro-inflammatory response. When we compare up top with the bottom, which is the addition of losartan, we have a less pro-inflammatory response and a higher anti-inflammatory response. Furthermore, angiotensin II induced the production of tumor necrosis factor alpha, increasing its concentration by 90%, an increase that was also blocked by losartan. All the previous results are consistent with the increases seen in EAE mice. As a summary, since there are no clear mechanisms for the pathogenesis of multiple sclerosis, the study aimed to demonstrate that angiotensin II contributes to the pro-inflammatory response of microglial cells in multiple sclerosis. The effects of angiotensin II were measured in EAE mice and in vitro by HM3 cells, and the levels of angiotensin II, IL-6, tumor necrosis factor alpha, INOS, ROS, and NO were increased, and these changes were blocked by losartan, which indicates that angiotensin II is the primary mediator of these effects. EAE mice showed similar changes and results. As conclusion, this study suggests that angiotensin II can upregulate the microglial cell pro-inflammatory responses, such as IL-6 and tumor necrosis factor alpha production, which may contribute to the pathophysiology of central nervous system inflammation in multiple sclerosis. Here are the references used for the introduction of this presentation. Thank you for your attention, and I hope you have a great day.